Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche. This lesson is brought to you by the Free State Department of Education in collaboration with Pagama Research and Development. So today we are meeting to do a revision for paper 1. So I hope you are ready. You are writing your preliminary examinations paper 1. And today we are going to be focusing on those aspects of every topic that is in paper 1 that is challenging or that we can focus on the tips that will help you to tackle your paper. Right, so some of the topics that we're going to look at include reproduction, meaning the human reproduction, and the response to the environment, both for the human and the plants, and then the endocrine system and negative feedback mechanisms. We are going to look at only those aspects that were found to be challenging in paper 1. So if you take a look at the topics that we need to cover for paper 1, they are like this. For term 1, the topics that you did were reproduction in vertebrates, which weighs 27%. That is going to be about, which weighs 5%. The first one is 5%. That is going to be about 8 marks of your paper. And then human reproduction, quite a big topic. So you see there's a circle around it. It weighs 27% out of the total paper there and giving us 41 marks. Then in term 2, you did responding to the environment by the humans, and it weighs about 36%. This is also quite a big topic, and the marks will be 54 out of 150. Remember, these marks are out of 150. And then in term 3, you also did responding to the environment by plants, and this weighs 9% and is going to give us 13 marks out of the 150. Then in term 1, overlapping into term 3, you did the endocrine and homeostasis. That is about 23% and gives us 34 marks out of 150. So all of these combined together will be your paper out of 150. Right, so how is the paper going to look like? I am sure that you have seen previous question papers as you were studying and also revising with your teachers. So you have seen the structure of the question paper for life sciences. The paper is going to be out of 150 marks and you are going to write for 2.5 hours, meaning you are going to get 1 mark per minute. So the way in which the question paper is structured for 2.5 hours, it is 1 mark per minute. Please use your time sparingly. Keep to time. Do not rush through the paper, or also do not waste time on questions that you may not understand. You can move on and also come back when you have enough time. Then the question paper is going to be structured in this way. You are going to find 2 sections, section A and section B. Then we all know section A is your short answer questions, your objective questions or your multiple choice questions. And you have your terminology questions and the matching column question. You can also have that 1.4 or 1.5 short question. So all of those questions will be 50 marks. That is 33.3% of the paper. And for section B, you are going to have a variety of question types. This section can also be further divided into questions of 40 marks each, or divided into 3 to 4 sub-questions. For example, 2.1 or 2.2, then 3.1 and 3.2. So these are all 4 sub-questions, but they must be divided into 40 marks each. All of them totaling 100. So this is where you're going to get your 50 and your 100. And please make sure that you obtain this first 50, because the first 50 puts you at a better position for you to pass. Because if you get 50, you are already on level 2. So please make sure that you get that 50, and then you can get additional answers here, and then it can put you at a better level. So that is how you should approach your question paper. Make sure that you kill section A. Right. So for the first topic that we are going to do is reproduction in humans. So we will look at, these are the things that you studied. So what DT exam guideline expect you to study. Remember, today we're not going to go into detail. We're just reminding you on those aspects that were challenging. So you need to look at the female reproduction system, all the diagrams, the functions, and all the processes, and the male reproductive system, and then puberty, also be able to explain what that is. Please know the events of the menstrual cycle and the hormones that are involved there, or the hormonal control of the menstrual cycle. I know most of you are struggling with the hormones. We'll find a way to help you remember the hormones. And then also the development of the fetus. So these are the aspects that you need to remember for this topic. Right. So we'll just get right into it. You're starting with the male reproductive system. Remember the male reproductive system, you need to know two views. There is a side view and a front view. Go through your question papers and check those diagrams. And every time you come across a diagram, make sure that you know the labels. So let us go through the labels quickly just to check and remind ourselves what is it that we find on the male. Right, we're going to start here on the scrotum. So the scrotum is this sac round about here. And then in the scrotum, we find what? We find the testes. Then the testes, on top of the testes, you find it. Remember, there are two testes. And then on top of each testis, there is an epididymis where the sperm is going to mature or be stored, and then it's going to go up. And as it goes up with this tube here, the tube's name is the vas deferens. And then this vas deferens is going to come into the three glands, but I don't want to talk about the glands as yet. Let us follow the route. Right, this is the vas deferens, and then after the vas deferens, you have the urethra, and then from the urethra you have your penis on the outside, and then you have the accessory glands. The three accessory glands are the seminal vesicle. Check how it looks like. The seminal vesicle will be that one that looks like a leaf of a tree, and then the prostate is a bigger circle, because there are two things that looks like a circle, but a bigger circle underneath the seminal vesicle will be the prostate, and then the last gland will be the corpus gland, or the last accessory gland will be the corpus gland, that is represented by a small circle there. So please make sure that you know the positions. And then the biggest one that you may not be asked about is the bladder, but in most questions you are not asked about the bladder. So that is just a review of the male. Now let us check these parts. So the male consists of the testes, which are situated in the scrotum. We've already seen that. And then the epididymis, that is on top of that. And then the vas deferens. And then the ejaculatory duct. So this ejaculatory duct will be this duct over here. Okay, let me just throw a dot on top of it so you can see where I've drawn a big red dot. That is where your ejaculatory duct will be. And then everything now leading to what? To the urethra, to the outside. So you see the urethra. Then the accessory glands that we've spoken about, your seminal vesicle, the prostate gland, and the corpus glands. Remember, you must know the functions of all of these. And then you have the penis that is on the outside. And then the seminal vesicles, what are the functions there? Let us remind ourselves the functions of the seminal vesicles is to produce the seminal fluid to transport the sperm cells and fructose for nourishment. So what is going to nourish the sperm is the fluid that is produced in the seminal vesicle. So this one that looks like a leaf, remember that it has fructose. Fructose is a type of sugar. So that one, it is going to nourish the sperm. And then the prostate gland. The prostate gland is a bigger circle there. It secretes a milky, slightly alkaline fluid that neutralizes the acidity of the vagina. Remember, the vagina is very acidic. So the sperm must come with something in its form that is going to neutralize that acidity. So this comes from the prostate gland. I hope you remember that. And then the corpus gland. The corpus gland is that one that we said is a small circle. It secretes fluid for greater motility of the sperm. So this sperm, remember, they have to move very fast. So what is going to make the sperm to move very fast? It is the secretion that is found where? Inside the corpus glands. And then the secretion of the prostate and the seminal vesicles, they nourish the sperm. There is a time where you're going to see that the functions are almost the same, or they are the same for the prostate and the seminal vesicle. If in Mind the Gap, you are going to see that they were actually indicated like that. Then it also enabled them to swim, and they provide protection. So that is your male reproductive structure. Then you are going to get questions. Remember, I said to you, you can look whether it's a side view or a front view. So for the sake of time today and having to cover all the topics that we need to cover, we are going to go through the questions together. So let us take a look at these questions or possible questions that you can get from the male reproductive system. Now, give the letters of all structures that are involved in each of the following. A, production of the fluid part of semen. So which parts are involved in this? So what you need to also note in the question is that they are asking for letters, and they are not limiting you. They say of all those structures that are involved in each of the following. So which structures are involved in the production of the fluid part of the semen? Right? So those will be A, B, and C. Why A, B, and C? All of these are accessory glands. And those accessory glands, we saw that whatever they are secreting is going to be part of the whole semen. Then, transportation passageway from sperm and semen. So the pathway of the sperm and semen. What are those parts that are involved in that? Let's check. You have D and E. So your D here is your vas deferens, and your E is your urethra. Remember, the sperms are going to travel from here, the sperm cell, and they are going to also collect or join here with all the fluids that are coming from these accessory glands, and they're gonna come out of the urethra. So the urethra is that is the passageway from the testis to the vas deferens to the urethra, then to the outside. So E and D are those parts that are responsible for that, or that form part of the passageway. Now name gland B. So B. Remember how I said you must remember them? This one looks like a bigger circle under the leaf one. So what will be the name of B? That will be the prostate. So I told you how to remember the name. Prostate gland. You're going to get 1 mark for that. Even if you wrote prostate, you're going to get 1 mark because the question has gland in it. Then name the hormone produced by the testis that brings about the development of male secondary sexual characteristics. So this is the male hormone. The male hormone, or the hormone that makes males to be males. So what is that hormone? It is secreted here in the testes. You have done this when you were doing endocrine system and also when you were doing this topic. So that hormone is testosterone. Remember we said because this word is long, you can cut it. Testosterone. Because when you write it, sometimes you write something else that does not sound like testosterone. So testosterone. Because when you say it quickly or fast, it might sound like something else. Right. Continue with the question. Test results show that this man has a low sperm count. Explain why a doctor would advise him against wearing tight underwear. So if he has a low sperm count, his doctor was going to tell him that stop wearing tight underwear. So what is the implication of that? Tight underwear will make the testis, or will put the testis close to the body. Remember, the testis are hanging outside the body and they're supposed to be -2 degrees Celsius temperature from the body temperature. So if you are putting the testis close to the body, you are putting it now close to the heat. Now, the temperature of the testes will be too high because they are hanging outside the body for a reason, for a purpose that they need to be 2 degrees cooler than the body temperature. So, but if you are wearing a tight underwear, you are putting the testes close to the body and making them to be too hot. And then the sperm, in that case, will not mature. Then sperm production will be negatively affected. So this is how you are going to answer this question about a man wearing tight underwear for his sperm count. 1.4. Explain what would happen to the male's ability to reproduce if the part labeled D is blocked off. So let's go back and check. This is part labeled D. This is blocked off. So will this man be able to reproduce if this part D here is blocked off? Now you are using your common sense. The answer is no. No sperms will be transported to the penis because remember, this is the pathway, and which will result in what? In no fertilization, and no offspring will be produced, and no reproduction will take place because everything now cannot go up. Remember, here we have sperm, but if sperm is in the testes and part D is cut, sperm cannot go anywhere. There won't be any reproduction. I hope that was easy for you also to answer. Then we come to the female. The female reproductive system also, there's a side view and a front view. Okay, let's write view there. Right, so you must know both. But in most cases, you will be asked on the front view, not to complicate things. And what are the parts? For you to get that 40% or that 50% strategy is for you to know the labels. You cannot remember other things, but please make sure that all your labels, you know them. Right. So we're going to start with the female. The female reproductive system consists of a pair of ovaries. When we say a pair, we say 2. So a pair means 2. So 1 ovary on the left, 1 ovary on the right. And these ovaries are attached to the fallopian tubes leading from each ovary. So you see here's a fallopian tube attaching with its fimbriae. This finger-like projection, we call them fimbriae. So it attaches on the ovaries of on each side, and then it leads away from the ovaries. So those are the fallopian tubes. And then they lead where? Into the uterus. This is the womb. The uterus. This is the part of that is called the uterus. You see this one here in the middle? Right. And then the uterus leads to the vagina. So this whole passage here is the vagina. Then before the vagina, there is a closing there on the uterus, is called the cervix. Now the cervix is the opening of the opening of the womb. So that is the cervix, then leading into the vagina, and then at the end of the vagina, the opening there will be called the vulva. So you have all the diagrams or the structures on your female. So there's a lot that is happening on the female, and we need to study that. So, right, okay, we have that. So we're going to explore. Right, the vagina is just a birth canal, and then we need to explore the sex hormones or the hormones that are produced here to maintain all the processes that are happening. And then we also need to check the transportation of the ovum to the uterus and implantation. So we're going to look at a lot that is happening here on the female. So let us go. So this is just questions on the structure. Remember, this is a strategy for you to obtain those marks. So you are going to get questions like this that may ask you to identify parts A and C. So let us take a look. What is A and what is C? So just at the end there of the uterus. Then your answers will be A is the fallopian tube and C is the cervix. I hope you got it correct. Then give the letters only of the parts where each of the following takes place: meiosis and fertilization. So where do you think meiosis is taking place here? Remember, meiosis or ovarian cycle, the ovaries needs to form, or the germinal layers needs to form here to form your ovum. And fertilization, that is where now the sperm cell fuses with the ovum, or the nucleus of the sperm cell fuses with the nucleus of the ovum. So where does that happen? Right. Meiosis will be here in the ovaries. And then fertilization will be at A, where? In the fallopian tubes. That is where the sperm cell or the nucleus of the sperm cell is going to fuse with the nucleus of the ovum and then forming that zygote. And that process, we call it fertilization, is going to take place here at A. Right. Continue with this paper. Stage 2 functions of part E. So part E will be here. So your part E, remember we discussed that part E is this one that is coming here. So what would be the functions there? Part E is responsible for the protection of the embryo. So the embryo is going to be embedded here. So it's going to stay in here for the full gestational period. So part E is going to protect that embryo from implantation to birth. So until that embryo becomes a fetus and it is born. It also provides space for the developing fetus. So this is the womb. This is the uterus. Think of the womb. Its role is to provide that space for the fetus and also provides protection, because it has so many layers in the endometrium. Protection for the developing embryo. Right. 2.4. Name the gland which secretes nutrient-rich fluid for the sperm to reach part A. So, nutrient-rich fluid, which part? So that the sperm can swim up to here and reach part A there. So, what is that nutrient? It is the seminal vesicles in males. So, remember we said it secretes a fluid that helps with the sperm motility, moving fast. So that fluid is secreted by the seminal vesicles in males. Right. Then you need to learn about puberty, what puberty is. So puberty refers to the age at which sexual maturity occurs and the sex hormones are produced, which leads to secondary male or female sexual characteristics and Gametogenesis, the formation of gametes. So, of gametes. So there's a lot that is happening at puberty. First, you are reaching sexual maturity. 2, sex hormones are now produced. 3, gametogenesis is happening, starting to happen. So that stage, we call it puberty. Now, what happens in males during puberty? Now, the pituitary gland starts to secrete FSH, which stimulates the testes to produce sperm cells. So the sperm cells at puberty, it's going to start in the pituitary gland to secrete FSH, follicle stimulating hormone, to go where? To the testes, to tell the testes to produce sperm cells. And then luteinizing hormone, or LH, stimulates the testes to produce hormone testosterone. So during puberty in males, you need sperm production and you also need testosterone to be produced. So all of these are under this influence, and these are the things that are happening during puberty. And then we know this hormone testosterone, once it is secreted, it is going to give us this secondary male or sexual characteristics in males, like your deep voice, face being beard, and then building muscles, and all of that, all of those secondary sexual characteristics that you are experiencing or that males are experiencing. And then with the females, and all of this, remember, we say they are caused by the hormone testosterone. With the females, starts when the pituitary glands secrete FSH. FSH is going to go where? It's going to go and stimulate a follicle in the ovary, or the formation of the follicles in the ovary to develop. And once they develop, they are going to secrete estrogen. Estrogen is what we refer as the female hormone. So that female hormone is secreted where? In the ovaries. But the ovaries are getting the message from where? From the pituitary gland, from the secretion of the follicle stimulating hormone. Then luteinizing hormone, which stimulates the development of the corpus luteum in the ovary to produce progesterone. So LH, luteinizing hormone, also plays a role during puberty to produce progesterone. So this one is not responsible for the second, sexual characteristics in female. Remember, estrogen is the one. So females, whatever they're experiencing in their body, growing calves, the development of breasts, all of those things, they are caused by the estrogen hormone. Unlike with the men, it is testosterone. With the females, it's estrogen. Right. So that is puberty in a nutshell. Then, another thing that is happening in the males and the females, we said during puberty there has to be gametogenesis. What is gametogenesis? Is the process of forming gametes. So the gametes in males are the sperm cells, and the gametes in females are ovum. So gametogenesis in males is called spermatogenesis. What is spermatogenesis? Is the formation of sperm cells. So this is a diagram that reminds you of how a sperm cell looks like. Sperm cell has a head. It has the head part, the middle part, and the long tail. So the head part consists of the acrosome, which looks like a cap, that contains enzymes to digest the walls of the ovum for fertilization. Then the nucleus contains 23 chromosomes, which carries the genetic material of the males. And then you find the centriole there. And then the middle piece consists of the mitochondria. Mitochondria is to provide energy for swimming. Remember, we said it had to swim fast. And then you have the long tail. Remember, when you draw it, when you are asked to draw it, the tail must be long. It must be longer than the head and the middle part, because then if it is not long, you're going to lose a mark. Then it is the flagellum tail, meaning it moves. Then it is used for swimming. Remember, they have to swim from the vagina into the uterus and then to the fallopian tubes. So that swimming is going to be helped by the flagellum tail over there. Right. So during the process of forming this sperm, what happens? This is what I want you to remember. You don't need to remember the whole process. Just remember this. How are sperm cells formed? Because that is spermatogenesis. Under the influence of testosterone, a diploid germinal epithelial cell. This is very important. Diploid germinal epithelial cell, which you find where? In the seminiferous tubules. The seminiferous tubules, you find them in the testes of the testes. Undergo meiosis and to form four haploid cells. This is the short and simple of it. You don't need to remember how many, what, or the long process. Just remember that in the testes, you have the seminiferous tubule that has diploid germinal epithelial cells that are going to undergo meiosis and form four haploid cells. That's it. That is your spermatogenesis. That is how sperms are formed. Right. And then this is gametogenesis in females, which is also called oogenesis or oogenesis. So oogenesis, you are forming an ovum. So this is a diagram of an ovum. Lately, you can be asked to draw also the diagram of an ovum. So this is how it looks like on your this layer here. You're going to have your jelly layer, which protects the ovum from drying out. And then you're going to find the cell membrane, which only allow one sperm cell to penetrate. The other name for that is the zona pellucida. So no, sperm cells comes in millions and millions, but only one sperm cell can fertilize the ovum. That is because of this membrane here, or the cell membrane here, because it's going to harden after fertilization has taken place, so that all other sperm cells can just go away or disintegrate or just go out of the uterus and the vagina. Right. So the cell membrane is going to allow for that. And then you have the cytoplasm, which nourishes the ovum. And then you have a nucleus that is also one end. So this is a haploid nucleus. It only contains 22 autosomes and one gonosome. So this is just a diagram of your ovum that you can be asked to draw. Remember the layers from outside to the inside. So how is this formed, or how is this ovum formed? So this is a diagram of an ovum. So how is it formed? Like in the sperm cell, you are going to follow where, when, what. So a diploid cell in the ovary undergoes mitosis here to form many follicles. So here we start with mitosis. We start at mitosis. Unlike with the males, the germinal epithelial, the germinal cells just underwent meiosis. But here, the diploid cell must undergo mitosis first. Remember mitosis, we are just replicating. Then to form many follicles. And then at the start of puberty, this happens from as soon as birth. At the start of puberty, and under the influence of FSH, follicle stimulating hormone, one cell in the follicle grows and undergoes meiosis. Now, you are going to say one cell in a follicle. So you're going to choose one follicle, one cell. And then that one cell is going to undergo meiosis. And when it undergoes meiosis, it is going to form what? A haploid ovum. So you'll see there will be others that are disintegrated, which we call them polar bodies and what. So they all die until you are left with only one haploid ovum. Unlike with the sperm where you have four, but with oogenesis in females, it's only one haploid ovum, because others just become polar bodies and they die. Right. So this is a question that you can expect on the sperm cell. The diagram shows a sperm cell. State how the following structures are suited to assist the sperm cell to perform its function: the mitochondria and the acrosome. So the sperm cells and the ova are produced by gametogenesis. This happens when a diploid cell undergoes meiosis to produce haploid cells called gametes. Tabulate two differences between gametogenesis in males and females, not mentioned in the text above. So whatever that you say must not be mentioned here in this text. So I would like for you to look at your worksheets and work on this question, and then discuss the answers with your peers so we can move to the next topic. So those will be your answers. Please discuss these answers with your peers. Then we come into the menstrual cycle. The menstrual cycle, you must just remember, it is under the influence of hormones. So what are these hormones? I know that you forget the hormones. And what is their sequence? If we can use this acronym here, we can call it an acronym, FOLP, then you can remember the names of the hormones. So the first one will be your follicle stimulating hormone. Everything must start with the follicle stimulating hormone. So this will be your first one. Follicle stimulating hormone. It is going to go into the ovaries and stimulate the development of the follicles. And then those follicles, what is going to happen? They are going to mature and mature until on day 14, one follicle is going to rupture under what? Okay, and then it's going to go to the ovaries, and then the ovaries are going to secrete the second hormone. What is the second hormone? Estrogen. So estrogen, so this will be your first one. Estrogen will be your second one. And then on day 14, luteinizing hormone is going to be produced. So luteinizing hormone becomes your L of the 4, 3, luteinizing hormone. And then lastly, progesterone is going to be secreted if fertilization did not take place. Check what is happening. The level of progesterone is going to go down. That is how you check. And then also the corpus luteum is going to disintegrate. I'm just trying to to remind you what are the reasons, or what do you look for when fertilization did not take place. But remember the sequence of the hormones. It's follicle stimulating hormone to the ovaries, and then the ovaries are going to secrete estrogen, and then after estrogen on day 14, luteinizing hormone is going to be secreted to stimulate ovulation. And then if fertilization did not take place, progesterone is going to be secreted. But if fertilization did not take place, the level of progesterone is going to decrease, and then you are going to start your period. Then let us take a look at the next graph. You are still following the same form, but here, fertilization took place because look at the corpus luteum. The corpus luteum is maintained. Why? Because it still needs to continue to produce the hormone progesterone. And also on the endometrium, or in the uterus, check here, the endometrium is thickening under the influence of progesterone. So everything is the same, but it differs when it comes to the progesterone. What is going to tell you that fertilization has taken place here? It is because the corpus luteum did not disintegrate. It is continuing to produce progesterone, and then the endometrium is also thickening. So those are the two aspects that you can look for. I know this graph can be challenging, but if you can just approach it this way, you will not go wrong. Right. And then fertilization, development of a zygote, and implantation. So let's start with this. Fertilization. What is fertilization? You see here the sperm cell is coming into the ovum, and only the nucleus. Oh, let me just cut it here, not there. The nucleus of the sperm cell is fusing. So fusion of the nucleus of the sperm cell with the haploid nucleus of the ovum. We call that process fertilization. And after this fertilization has taken place, a zygote is going to be formed. So let us go to zygote. So a diploid zygote. Why is it diploid? Because it consists of the cells from the mother, or from the female, and from the male. So it has 46 chromosomes. Hence it is diploid. Then this zygote is going to undergo mitosis. So it's going to start by becoming 2 cells, then 4 cells, then 8 cells, and then it becomes a morula. Please check the sequence. There's a song on YouTube that you can go check that sings about the development of this zygote. Right. Morula, now it is a ball of many cells. So you see there are many cells. And then it becomes a blastula when there is a hollow inside it, or there is a space inside it. So it has grown to this. Now this blastula is going to come through into the fallopian tube, and then it's going to do what? It is going to implant itself, or attach to the endometrium. That process of attaching to the endometrium, we call it implantation. And then from implantation, you can see it here how it embeds itself. They've just made this picture to grow, or to zoom it out here. So when it implants like this, therefore it can develop into an embryo, and then up to it develops to a fetus until the baby is born. Now the fetus has grown, and it has attached itself to the uterus. So let's just say here. So we are taking it from the previous slide. So once it has attached itself, you are going to do what? Check what are the functions of all these layers surrounding our fetus. So the most important will be your chorionic villi. So your chorionic villi are these finger-like projections. You see, they are embedding themselves or entering the uterus during that process of implantation. So they formed a placenta, and that placenta has chorionic villi. So those villi are the ones that are entering in between the spaces of the blood, or projecting into the blood spaces of the uterus of the mother. Then it is going to provide maximum contact with maternal blood, and then it also forms part of the placenta. That is your chorionic villi. So this one that is entering there. And then you have your umbilical cord. I know that all of you know the umbilical cord because it's that one that they cut once the baby is born. So what is the function of that umbilical cord? The umbilical cord is a flexible cord containing 2 umbilical arteries and 1 umbilical vein. So if you check here, there are 2 arteries and 1 vein. Now, the arteries carry carbon dioxide while the umbilical vein carries nutrients. So the baby is going to get the carbon dioxide, is going to go out of the baby through the arteries, and then the vein is going to bring nutrients to the baby. Then you have the amnion. See, this is the covering of the amnion, which secretes amniotic fluid. And this amniotic fluid, this is where the baby is swimming in, and they are drinking and eating it and doing whatever. Protects the developing fetus. So it cushions the baby around as the mother moves. So the amniotic fluid acts also as a shock absorber and protects against temperature changes. So even when it's cold outside and the mother is not putting any blanket, the baby is protected because this protects against temperature changes on the outside. It also protects the fetus against dehydration because it is a fluid. So everything on the baby is, or on the fetus, because this is not born yet, on the fetus is moist, it's controlled, or it is protected against dehydration. So those are some of the layers that you need to study during or when the fetus is developing in the mother. Right, and then the last one will be your placenta. I know that you also know this one, the placenta, because you know that the mother has to also give birth to it after giving birth to the baby. The placenta's function is for gaseous exchange between the mother and the fetus, providing also nutrients to the fetus, and it also secretes progesterone. So it keeps to maintain and also gives nutrients and also for gaseous exchange. So it plays so many roles there. That is your placenta. Different types of questions that you can get. Please work through this question also with your peers and understand how the placenta is formed and those functions there. Also continuing, taken from KwaZulu-Natal Prelim Paper 1 of 2022. But you have this in your worksheet. Right, so let us go to the next topic. Now, the next topic will be human endocrine system and homeostasis. Right, this topic, you just need to know the endocrine system as a whole. Remember the endocrine system, remember that diagram of a human torso with all the areas where the hormones are secreted. Yes, so you must remember all those glands and their hormones and the functions of those hormones and negative feedback. So we're just going to look at this diagram. The diagram, remember the pituitary gland secreting 5 hormones. 1, 2, 3, 4, 5. Growth hormone, thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, and prolactin. Then you have the hypothalamus, which plays a role in temperature regulation, secreting ADH and osmoregulation. Then you have the thyroid gland secreting the hormone thyroxine. Then you have the adrenal glands secreting the hormone adrenaline. And adrenaline plays a role also in the flight or fight response. It makes your body to do all of this. Increase the heartbeat, raises blood pressure, speeds up the conversion of glycogen to glucose, causes the pupil to dilate, all of this flight or fight. Then you have aldosterone secreted by the adrenal glands on top of each kidney. Then we have insulin secreted by the pancreas by the pancreas and the islets of Langerhans cells, and glucagon also secreted by the pancreas. Then you have your ovaries secreting estrogen and progesterone, and then there would be 2 testes that are secreting the hormone testosterone. And I take it that you remember the functions of all those hormones. If you don't remember, please go through the functions. Remember negative feedback. Negative feedback consists of 3 main components. The first one, there should be a receptor receiving what is happening in the body, then detecting the imbalance. Then there should be a control center, what is going to respond. And then there should be an effector, where the response is going to be. So an example of this person of a negative feedback can be this person here under normal. You see this person is happy. And then when the person's body temperature started to decrease, the person started shivering. And then negative feedback mechanism is going to do what? To control this and make sure that our person's body temperature returns back to normal. It is going to increase it until it turns back to normal. Or another way can be this person's body temperature increases a lot and the person starts sweating, they are heating up a lot. The negative feedback mechanism is made in such a way that the body temperature must decrease until the person's back to normal. So that is how it works. Remember the sequence of events. Step 1, an imbalance should be detected. A control center must be stimulated. So what must be stimulated after an imbalance is detected? Then the control center must respond. Then after that response, that message must be sent to the target organs. Where? And that message must tell the target organ to respond. Once it receives the message, it will respond accordingly, and then it opposes or reverses the imbalance. It will oppose what was happening at the first, whether it was high or low. It will oppose it accordingly, and therefore balance is restored. Remember all these sequences whenever you are answering a negative feedback question. So the negative feedback that you need to study is that one of the thyroid. So you'll follow that. You have this in mind, the gap. Then the next one is of blood glucose. And then thermoregulation. Then you're going to get questions on the responses there. Please go through this also. The answers don't mean anything if they do not make sense to you. Just to recap on that diagram, you can also get questions like this. This was taken from the Free State Prelim Paper 1, 2022. So this was a paper that was written in the Free State. The diagram alongside represents the human endocrine system. So this is this diagram. Write down the letter and the name of the gland that produces a hormone that controls the growth of long bones. Now this one, any Tom, Dick, and you must get it right. Please check. They say a letter and a name. You are going to lose marks if you don't pay attention to that. You see, it says 2 marks. But if you are just going to write a name only or a letter only, you might lose marks because sometimes some of the marking guidelines want you to write both for you to get these 2 marks, or you might get only 1 mark. So please be careful when you are answering such questions and always look at your mark allocation. So what would be that hormone? That will be a P2A, which is a which is the pituitary gland. So this letter, the pituitary gland secretes hormone, growth hormone, because the growth hormone is the one that is responsible for the growth of long bones. But here they just wanted you to write the letter and the name of the gland that produces that hormone. So that is A, your pituitary gland. Then that is how you get your 2 marks. Gland E is only found in males. So let us go look here. So these are only found in males. Explain the consequence if gland E cannot secrete its hormone. So gland E secretes testosterone. Then if testosterone is not secreted, remember the functions of testosterone. No development of secondary sexual characteristics in males, and no sperm will develop, or sperm count will be low. So we need that hormone for all of these to happen. Right? Describe the homeostatic control if the salt level in the body are low. Remember, salt is also one of the homeostatic controls that you need to study. Also water, right? So salt, this is the whole story. I don't want to go through it just to save time. Now, name 2 other substances or factors except salt that should be kept constant in the internal environment of tissue fluids. So meaning all other substances that should be kept constant except for salt. So those are the ones that you've studied. Water should be kept constant. pH should be kept constant. Carbon dioxide, glucose, temperature. So you can get any 2, but check here, you're going to mark only the first 2. Very important also this, because sometimes you are asked for 2 things and then you write 5, but you find that the first 2 are incorrect and then number 3 is correct. The marking guideline stops the marker here from marking the first 2, irrespective of whether the ones that are coming after that are correct. So please be careful. When you are asked for 2, be sure of the 2 that you are writing, especially the first 2. Right, so this is another question that you can practice with in your worksheet. It was taken from Mpumalanga Prelim Paper 1 for 2022. Right. Continue with that question. Right, so I would like us to go into the next topic that you need to highlight things on, that is the plant and human response to the environment. So let us check. What does the exam guideline say you must know here with the plants? Functions of the following hormones. We have done these hormones with you. Remember, it says you must know the functions of the auxins, gibberellins, and abscisic acid. The exam guidelines also says you must know the role of auxins in geotropism and phototropism. The role of the following as plant defense mechanisms, chemicals and thorns. Do you remember how plants defend themselves with chemicals? How plants defend themselves with thorns, right? You must also remember, now this topic is about 13 marks of paper 1. So phototropism and geotropism. Tropism from the exam guideline is a growth response which a plant makes with respect to a directional stimulus. So this one, tropism, does not say which one. So it is the general term. It is the general one if light is not mentioned, if gravity is not mentioned. So what can be those stimulus? The first stimulus can be light or gravity. These are the 2 that you are expected to study. So this plant here, potted plant, shows you that the stem of this plant is showing phototropism. However, the roots are showing you geotropism. They are growing towards gravity. The stem is going towards light. Then it can happen in the same way. You need to look at the concentration of the auxins in each instance. This is what you must remember. whatever diagram that you must get, remember that the auxins, when a plant is facing unilateral light, the auxins are gonna go where? Are gonna go to the darker side, because light destroys the auxins. And then on the darker side, they are going to stimulate the growth. Hence, the plant is gonna bend towards the light because of a cell elongation that is happening on that darker side. This is what you must remember about the auxins. And then in geotropism of the shoot, remember the shoot can be a stem, the upper part. The auxins are gonna come to the gravity. Remember, the gravity attracts the auxins. And then where the auxins are, they stimulate growth. Hence, the shoot is gonna grow upwards. So you need to study your plant exactly what is it that you have. But in the root, the story is different. The auxins are gonna come to the gravity side and they're gonna grow. They're gonna cause growth or cell elongation on the gravity side, and then our root is gonna grow towards gravity. So if your plant is placed in a horizontal way, so you must just check how your plant is placed and check where they are asking you or what are they asking you about the auxins. Right. Okay. Going to the next slide. Right. So this is a table that is summarizing what we were just talking about. The role of auxins in phototropism and in geotropism. When the stem is exposed to unilateral light or light coming from one side only, the auxins concentration will be high on the dark side. So we did say, why is it on the dark side? Because light destroys the auxins. Then more growth occurs on the dark side because auxins stimulate cell elongation. Because auxin stimulates growth on the dark side. And then the stem is going to bend towards the light. In geotropism, when the root is placed horizontally, very important, you must check where the light is coming from and how your plant is placed. If your plant is placed horizontally, like this. Now, my plant looks like a person, but it's a plant. Right. Only one side is exposed to gravity. So this side, the lower side, will be the one that is exposed to gravity. The auxins concentration will be high on the lower side. So they will come here, the auxins, on the lower side of the root. Because gravity attracts the auxins. More growth occurs on the upper side. So on this upper side of the root. Because auxins on the lower side inhibit growth. So where the auxins are, on the lower side, they will not be able to work as normal. They are going to do the opposite, which is to inhibit growth, meaning it stops the growth on the lower side, and then the upper side is going to grow more. Hence, our shoot is going to grow towards light, and then the root is going to bend downward. Not light, I wanted to say gravity. And then this is one of the typical examples that you can get on plant response. Please check what you are asked about the auxins and check how your plant is faced. So here you see it is placed horizontally. You can already think of geotropism. There is no story about light. And then if you see something about light, then you will see the rays of light coming, and then you have to answer accordingly. Right, please do this activity also on the roles of the auxins in geotropism and phototropism. Right. So, other plant hormones that you're supposed to study are gibberellins and abscisic acid. I hope you still remember their functions. Gibberellins is responsible for stem elongation, the germination of seeds. Right. Elongation, the germination of seeds, and it promotes flowering. So I wanted all of this to come together. Stem elongation here, the stems are elongated. The seeds, germination of seeds here, and then promoting flowering. Remember, these are your gibberellins. And then abscisic acid, remember we said this hormone is that winter hormone. This hormone is an inhibitor of growth. What does it do? It causes the plants to become dormant in winter. And the lack of water causes the stomata to close when the plant wilts, when there is not enough water. So remember this diagram, it is orange and brown as it is, it reminds you of winter or autumn. Then think of the way the hormone there, which is abscisic acid. Then this is what it causes. It causes the stomata to close. So this is a closed stomata, just to retain the water. Remember, the stomata, you find it on the leaves. So you don't want the plant to lose more water, because then it will wilt. So that is your abscisic acid. Abscisic acid, winter hormone, inhibits growth or stops. To inhibit is to stop or to prevent. Right. So that is on the plant response. Let us do the human response now to the environment. Quite a big topic also, because it has so many subtopics. So this is what you need to study: the brain and all its parts, the cerebrum, the cerebellum, medulla oblongata, the hypothalamus, and the corpus callosum. So you need to know all these parts and their functions and where they are in a diagram. Then you need to understand the spinal cord, the structure of the spinal cord, and describe reflex action using a diagram. So I hope you all remember that. Then you must also know the neurons and your 3 types of neurons: your motor, sensory, and interneurons. How do they look like? How can you differentiate between them and their functions? And then the human eye, all the parts and their functions, and accommodation and pupillary mechanisms. These are 2 processes that are happening in the eye. You must be able to describe them. And the human ear, the structure and the functioning of the parts of the human ear, and then the process of hearing, the process of balance and hearing defects. Also here, visual defects. So you must know your cataracts, and here you must know also your how when to use the grommets, hearing loss, and also hard of hearing, and when to use the grommets, when to use all these other aids. Right. This topic is 54 marks of your paper 1. So you need to spend a lot of time on this topic. Right. So the nervous system, remember we said the nervous system is divided into the central nervous system and the peripheral nervous system. So let us start with the central nervous system. The central nervous system is very easy. It is just like it's a central. It starts with the brain and the spinal cord. So that is your central nervous system. And then divided also into the peripheral nervous system. But the peripheral nervous system is all these other branches of your nervous system. So the ones that are coming to the side to the rest of your body, except for the brain and the spinal cord. All these other nervous systems that you find here are your peripheral nervous system. And it can be divided into sensory pathways and motor pathways. Now, the motor pathways can further be divided into the somatic or the voluntary nervous system, which is under your control, but not controlled by the brain. And then the autonomic nervous system, which is involuntary. And then the autonomic nervous system can further be divided into the sympathetic division and the parasympathetic division. So these are the ones that are working during the flight or fight. It is autonomous. Right. So this is your brain. Remember, forms part of the central nervous system. You need to remember the largest part of the brain is the cerebrum, thought processes and all that. And then the corpus callosum, dividing the 2 hemispheres or the brain into the 2 hemispheres. the right and the left, and then the hypothalamus playing a role, we just saw there, in temperature regulation, and then your cerebellum working with balance, remember balance, cerebellum. Then your medulla oblongata, breathing. So those are the things that can help you to remind you of all other functions of these parts that you need to study. And then your pituitary gland, or the hypophysis, we know it is responsible for secreting all those 5 hormones that we mentioned when we were doing endocrine system. So this is your brain in a nutshell. So you can go through all those functions to remember them. But remembering the parts, very important, because you cannot miss a part, or you cannot miss a label. Then this diagram of this person here shows you what I was talking about, the peripheral nervous system. So anything else but the central nervous system. So anything that comes on the side forms part of the peripheral nervous system, and it is divided in that way which we saw there. And then a reflex arc. So this is the pathway of your impulses until you react. So the reflex arc, what does it have? So it will start from the receptor. Your receptors will receive stimulus, and then that stimulus will go through your sensory neuron. Okay, right, we missed another label there. Right, this is your sensory receptor. So receptor receives the stimulus. So if you forgot which one is the receptor, which one is the effector, just remember like this: receptor, receive. And then it receives the stimulus, and that stimulus is going to go through the sensory neuron. The sensory neuron that goes through the dorsal part of the spinal cord, or the dorsal part, or the upper part, and then it's going to go through to the interneuron, and then that impulse is going to now travel from the interneuron to the motor neuron. Okay, they are coming so slow. Through the motor neuron of the ventral part of the spinal cord, and then to the effector. Effector is where you feel the effect. So you feel the effect on the effector, and then you start acting accordingly. And you must remember, this reflex arc, it does not include the brain. The brain does not play a part here because it is very rapid, and you don't have to think about it, because if you put your hand on the stove, you have to quickly move and react. But if it has to go through the brain, you'll have to think about it. Think whether you are burning or you are not burning, and then you want to remove, and all that. It will be slower. So the reflex arc is very quick and rapid, or the reflex action rather, because the arc is just a pathway. The action that you are going to make through this arc is very rapid. And please check the differences between those two. Reflex arc and reflex action, not the same. Then your diagrams, I hope you know all the labels, but let us check. How do you know that this diagram is a motor neuron, or is a sensory neuron, or it's an interneuron? First, you must check. The last one will have a receptor cell. So this is where you are going to have your receptor. No, the first one, sorry. The first one. So this is your first neuron, which is your sensory neuron. It is what you sense the stimulus with. So it's going to have a receptor cell where you receive the stimulus. So there are no dendrites or terminals on the other side. And another thing that you can check to know that this is a sensory neuron, the cell body is in the middle, on the side. Let's just write on the side, yes. And then the second one will be your interneuron. What is your interneuron? So this will be your second neuron. The interneuron, its dendrites must be able to meet with the terminals of the previous one so that there can be that synapse and transfer of impulses. So this one, as you see, is going to match here. You see, then there will be that transfer of impulses. And then also here, the cell body is in the middle. Yeah, here the cell body is in the middle. The cell body is in the middle. Here, the cell body is on the side. Cell body is in the middle. The middle. So everything of the inter is in the middle. And then the last one, your third one, will be the motor. The motor, remember the terminals of the axons are going to meet with what? With the dendrites. But here the motor, look at the cell body. The cell body is inside the dendrites. See? Inside dendrites. So everything must flow. So from the first one to the second one to the third one. And also check the labels. So the impulse will be from this side, from this direction, top to the bottom, to the top here, to the bottom, to the top here, to the bottom. So that is how it's going to flow, and that is how you're going to know which neuron is which. Then this is a question that I would like for you to go through with your worksheet that you have there. Then continuing with that question. Excuse me. Okay, another one of John. Right, so I would like for us to look at this question because questions with this such wonderful diagrams, they are testing your knowledge of what we've just discussed, that how are you going to collect marks. So the diagrams represent two types of neurons, diagram 1 and diagram 2. So identify them. What is diagram 1? Using the knowledge that we've just given you now. So what would you say is diagram 1? Is it a motor? Is it a sensory? Is it an inter? Remember, look at where the cell body is. The cell body is inside the dendrites. So that is going to be what? Your motor neuron. And then this one, the cell body is on the side. What is this one? It is your sensory neuron. Then identify part C. Part C, we've already said it's a cell body. Now give the letter and name. Pay attention to this type of question. Letter and name of the part that provides electrical insulation. So that will be your myelin sheath. Remember, these neurons are transmitting electrical impulses. So we don't want them to shock or to have electricity there shocking you. So what is going to provide that insulation is what is covering here, which we call it the myelin sheath. So myelin sheath will be letter B. So you give the letter and the answer. The one that receives incoming impulses. So from the previous neuron. So which ones are receiving those incoming neurons? What is this? Incoming impulses. Those will be your dendrites. A, here is A, indicating dendrite. So you see how important it is for you to know the functions, the labels, and the functions and the structural suitabilities, because then you put yourself in a better position to answer questions, even not on a deeper level. Then which neuron, 1 or 2, transmits impulses to the central nervous system? So which one takes these to the central nervous system? So that will be diagram 2, which is your sensory neuron, because it takes it from the receptor to the CNS, through the spinal cord, because without it, you will not be able to sense anything from the external environment. Right, so this is another diagram that you need to know of a reflex arc. Remember, without even looking at the question, once you see a diagram like this, just remember that look at the arrows, because they are also leading you to say the stimulus is coming in. Then if it is coming in, this must be my sensory, because it is the one that is going to receive those impulses and transmit them to the central nervous system, into the interneuron, and then to the motor neuron, and then to the effector, without even looking at the sensory neuron, interneuron, and motor neuron, and then effector. This is your receptor. So once you've done this, whatever question is going to come up here, it will be easy for you to answer because it shows that you understand the diagram. This is what it means when they say study the diagram. Right. So you will answer the questions based on this diagram. So please make sure that you tackle your diagrams. You don't enter any exam without understanding all the diagrams. Then we come to the structure of the eye. This is the external structure of the eye. Remember, with the eye also, you need to know all the functions and all the parts. Remember, the easiest way to study functions that are scattered around like this is to go through the eye layer by layer, from the outside to the inside, until you have covered all your labels. So your labels will be your sclera, and then you come to the front, your cornea, then you come inside the aqueous humor, the pupil, then you have your muscle. You see, now you are building up your eye from outside into the inside, also remembering your functions here. So please go look at these diagrams, go look at these labels, and remember what they do. What are their structural suitabilities also? Because some of the questions that may come from here are questions of structural suitability, to say, how is the sclera suited for its function, or how is the lens suited for its function? So those things of being transparent, being what, very important. So as you study the diagrams with its labels and functions, also study the structural suitabilities where appropriate. Then another process that you need to study with the eye is the process of accommodation, and that one of pupillary mechanism. Accommodation is for near and far vision. Pupillary mechanism is for light and dim situation, where you are in a dark room or in a light or in a bright light situation. So that is what you are going to explain. The accommodation deals with the lens. Pupillary mechanism deals with the pupil. So please remember those. So during accommodation, I would like you to use this acronym to remember the processes. Accommodation is the ability of the eye or the lens of the eye to change its convexity to see objects that are near or far. So the eye has that ability to change the convexity of the lens. So that is accommodation. Then what happens when an object is less than 6 meters away? Meaning when the object is near. Once they say less, you say this is near. I want you to write this. Very important. Please write it down. CC, SS, TD, LM. This is what is happening when an object is less than 6 meters away. What is CC? The ciliary muscles contract. You are going to get a mark for that. Please, please, please don't lose these marks. Make sure that you collect. CC, ciliary muscle contract. When does that happen? When an object is less than 6 meters away. Then the second statement. This one is not so important. The suspensory ligaments slacken, or they loosen, but you use the term slacken so you can remember. SS. Suspensory ligaments slacken. That is your number 2. You get a mark for that. And then the tension on the lens decreases. Tension on the lens decreases. That is your TD. And then the last one, the lens becomes more convex. Lens becomes more convex. Meaning when something is close, the lens is more convex. Then the refractive power of the lens increases, then a clear image is formed on the retina. Do not forget this statement, because also this is a concluding statement that is going to give you a mark. Then when an object is more than 6 meters away, meaning it is far, what is going to happen? Also have your acronym. You see? CR, ST, TILL. I hope you are noting them down and writing them. So in this case, unlike with the first one when we had CC, when it is far, it is CR. The ciliary muscles relax. Ciliary muscles relax. Then the suspensory ligaments become taut. ST. Suspensory ligaments are taut, meaning they are tight. Or you can just write tight because at least it's a T again. So the suspensory ligaments are taut. Then your next TI. Then the tension on the lens increases. So the tension on the lens is going to increase. Then the last part, the lens become less convex, meaning now our lens is like this. You see? When it's far, the lens is less convex. When it's close, the lens is more convex. Then the refractive power of the lens decreases, and then a clear image is formed on the retina. A clear image is always going to be formed, whether it's near or far. So always close your statement with this. So I hope for accommodation you'll be able to get those marks using this. Then for pupillary mechanism also, we have CC, if this is the last thing that you can write before you leave, CC, RR, PC, and LL. Go figure them out what they are. Your CC, you start here. The circular muscles are going to contract, and then your radial muscles are going to relax, and then your pupil is going to constrict, and then less light is going to enter the eye. So you see, so please use them. Use Mind the Gap and also use this acronym to summarize the processes and make them easy for you to understand. So CC, RR, PC, LL. That is for bright light. So this one is for bright light. And then for dim light, it is CR, RC, PD, and ML. Please study them so they can help you get those marks. Then these are questions on the eye and pupillary mechanism and accommodation. Please practice as many questions as you can for this. And then the last part is the structure of the ear. So you need to understand the ear is divided into 3 parts. So the outer ear, the middle ear, and the inner ear. Remember, the outer ear consists of what? The pinna. And you must remember the functions and the structural suitabilities. And then the auditory canal. From the auditory canal, you now start getting the middle ear, which starts with your tympanic membrane and your 3 ossicles. Ossicles are those small bones, your malleus, incus, and stay. And then you have your oval window and your round window. And then you come into the inner ear where you have your semicircular canals, those ones that look like semicircles. And then the one that looks like a snail is your cochlea. And then the one that is living like a nerve is your auditory nerve going to the brain. And then in the middle ear here also you have the eustachian tube. Remember the functions of all these parts. Then also the last thing that we can follow is the path of sound. How do you end up hearing? Where does sound start? Where does it start to become vibration? Where does it become pressure waves? And when does it become a nerve impulse? Because this is very important when you answer your questions. So from the outer ear, the sound waves are going to start coming through the pinna into the auditory canal, through the tympanic membrane or the eardrum. Then we enter the middle ear. And once they are at the tympanic membrane, we call them, they start to become vibration, because the tympanic membrane is going to vibrate. Vibrations. Yeah. Then those vibrations are going to move through to the ossicles and causes the ossicles to vibrate even more. And then the oval window is going to do what? To make it, to amplify those vibrations. And then you come into, remember the oval window is between the middle and the inner. Then those vibrations come into the cochlea, and then there is the endolymph and the perilymph, the fluid there, and then goes to the organ of Corti, the special organs that will allow you to hear. And in the inner ear, once they start reaching the cochlea, we start referring to them as pressure waves. And then when they go to the auditory nerve, they shall have been converted into nerve impulse, because the auditory nerve can only transmit impulses to the brain, because the brain deals with impulses. That's where they'll be able to be interpreted as sound. And if it is sound, remember, it will go to the cerebrum. But if it is for balance, it will go to the cerebellum. Yes, right. So these are just the parts that show you. This is what I want you to remember. Very important. How does the ear maintain balance? Through acids and sump. Write those down. Acids. The structure there is the ampulla and the semicircular canals. Where do we find this? Or which receptors are those? The cristae. And then this is when the direction and speed changes. And then when the position of the head changes with regards to gravity, the receptor that is going to react there is the maculae. Macula. Where do we find the maculae? On the circulars and the ultriculars. So SUMP will help you remember that it is for position of the head. Acids will help you remember it is when there is a change in direction and speed. So which receptors are responsible, and where do you get those receptors? In the structures. So remember the structure and the name of the receptor, and when do they react. So that is the best thing that you can remember on part of balance. Then these are just the questions on the ear. Remember, you have your structure, very important. Your semicircular canals, you already see your top one is your round window, oval window. You see your cochlea, you see your optic nerve. Study your diagram before you even answer the questions. Write the labels here so that when you come to the questions, they are a bit easier for you to answer. So, okay, those are the answers that you can go through. Go through as many question papers as you can. Paper 1, you will get the same topics that we've discussed today for paper 1. So this is still ear, hearing, and balance. Know your labels. Know the functions. Know the structural suitabilities. Right. So once you go into the exam room, what is it that you need to take? So you need to have your pen, whether a blue pen or a blue pen or a black pen. Most preferably a blue pen. You must also take a pencil with an eraser, or a pencil and an eraser separately, but you must have a pencil and an eraser. You must have a ruler. You must have a compass to draw your pie chart if necessary, or if expected to do so, and also your protractor to help you draw that. And don't forget a calculator. Very important, because there will be questions that are asking you for calculations. So for you not to panic, please make sure that you have all the necessary utensils or equipment to write your paper. And I would like to say good luck for your paper 1 until we meet again. And thank you so much for watching this lesson. See you again next time when we do paper 2.